Hello, everyone, and welcome to the LINX Distinguished Speaker Series. My name is Narani Nimpuno, and I'll be moderating today's session. This is a live webinar, so welcome to all of you who are participating live, but even to you watching it on YouTube afterwards. Those of you who are watching it live, do make sure to pop your questions in the Q&A. We like it when it's interactive, and I'm sure you have a lot of questions for to today's speaker. So the LINX Distinguished Speaker Series is a series of talks gathered here at LINX with experts in the industry who have deep knowledge and expertise and often a unique perspective on a particular subject. And today we have a real treat for you. We have Bert Hubert, who's going to present how life, included COVID-19 vaccines, is digital. And because it's a cross-disciplinary subject, I actually invited a friend and medical journalist, Bert Hubert, who will be helping me with moderating this session and will hopefully ask some more insightful and competent questions that maybe I'm capable of at the end. So thank you, Bert, and thank you, Bert, for joining us today. So first, a few words about Bert Hubert. I think he's familiar to some of you in this industry. Bert is the founder of PowerDNS, software that powers a significant fraction of the internet. In addition, Bert co-founded a joint venture with notable security company Fox IT. In between, he spent several years working for the Dutch government on cyber and national security. After selling both companies, Bert spent 18 months doing DNA research at TU Delft, leading to publication in a major science journal. These days, he focuses on open standards, decentralized communications, internet measurements, and research, mostly DNA and GNSS. In addition, he sits on a government board. So with that introduction, I won't delay this anymore, and I will hand over to Bert to give his great presentation. Bert, the floor is yours.
Okay, thank you very much. I'm going to attempt to share my screen. I hope that works. Always a little bit exciting. Okay, can you see my presentation now?
Yes, we can.
Okay, very good. So welcome everyone. This stuff it's jam-packed, so I'm gonna skip a lot of stuff just to get to the good stuff. So welcome. Let's see if I can. So I will skip my introduction with one small exception. So I'm sort of a gaslighting guy. I come from the internet. I do all kinds of things, and here I'm talking to you about DNA. And you may wonder, can we trust Bert? Does he know what he's talking about? And you should definitely be careful before you trust me. But I have somehow managed to fool a major journal into publishing my DNA research. So there is a chance that I know what I'm talking about. And I spent 20 years thinking about it and doing research. So I hope it's good. So DNA, life, is it digital? DNA is like the tape drive of life. It's very robust. We routinely restore DNA from 50,000-year-old remains, which is how we have a copy of the complete Neanderthal DNA, for example. And unlike computers, DNA uses 4 letters, A, C, G, and T, whereas computers use 0 and 1. And interestingly, even the original DNA papers already called DNA digital. And they actually called the 4 A, C, G, and T letters, they called them digits. So it's a very digital thing. And one of the most mind-blowing things about DNA is that life has been around for a while now, over 4 billion years, as far as we can tell. And you can take DNA from any part of the animal and a whole living, the whole nature, everything in nature. You can take one piece of DNA, put it in another animal, and it will work. So there is just one standard for DNA on this entire Earth, which is sort of stunning because I cannot even play a CD anymore from like 5 years ago, whereas life has managed to retain compatibility for billions of years. So that's already very impressive and better than we do. So how do we know this stuff? If we look at all forms of life, they fall apart in 3 kingdoms. There are actually 2 completely different kinds of bacteria, and they are completely different. And we know they have been different for at least 4 billion years now. And One day, these bacteria and archaea, as they are called, they joined up and they made a more complex cell, and that is us. So we came from the marriage of these very ancient two kinds of bacteria that still are somehow still with us. And one of the more interesting things that we know that life started around 4 billion years ago, and we know that water appeared on the planet 4.2 billion years ago. So in those 200 million years, life already got going. And you see this very pretty vent, and this is the current thinking about where life might have originated, and which is deep in an ocean vent where volcanic plumes arise full of interesting chemicals and heat. So that's where we think it all came from. But again, it's 4 billion years old, and this is a 4 billion year old software project. So some fun DNA facts. As storage goes, DNA is even more impressive than microSD. It requires 0.3 nanometers per letter, and that's 40 atoms. And that's not a lot. You could not make it a lot less than 40 atoms before you get really deep into all kinds of quantum effects. So maybe there would be a better way, an even tighter way of storing data using maybe 20 atoms, but 40 is probably extremely close to the physical information density limit. And also it doesn't weigh anything. So if you have 1 billion letters of DNA, it weighs 1 picogram. And so that is like really nothing. And you should realize that every human cell contains over 1 gigabyte of DNA. And that all fits in the cell, and even it's only a tiny, tiny part of the cell. It is also super redundant, and it's very impressive. And briefly, what it looks like. These are the 4 characters of DNA. So there's thymine, adenine, cytosine, and guanine. And so these are the T, A, C, and G as we know them. And what not many people know is that the G in guanine actually stands for the guano from bat droppings, which is where it was first isolated. So maybe good to know that if you see a G in DNA, you can think about bats. And T and A are very attracted to each other. So whenever there's a T in DNA, it would love to get close to an A. And whenever there's a C in DNA, it would love even more to get closer to G. So it's very important to realize that these pairings, these 2 pairs are what powers DNA in everything. So how can you look at this? Every character of DNA is like 2 bits. So we can say arbitrarily A is 0, 0, C is 0, 1, G is 1, 0, and T is 1, 1. And you may now think that, yeah, that's not fair because my computer actually runs on ones and zeros, and you are running on this guano batshit molecule. And that's not the same thing. And then I'd like to remind you of this. In nature, these bits of DNA are these molecules. But if you look at how we store ones and zeros in computers, that's actually also a physical thing. So your computer does not process ones and zeros. It processes voltages and currents and charge densities or north-south magnetic transitions. And so if you look into it, it is entirely fair to say that if computers are digital with their voltages and noises and frequencies and charge densities, if we call these things ones and zeros, then it's entirely fair to also call these molecules digital storage of information, being A, C, G, and T. And I'm going to skip this one because we're really going to run out of time. But the good thing to remember is this DNA stuff is extremely redundant, and which is why it can last 50,000 years easily enough. So by now you might think, wow, nature, that design that nature built is like just stunning. It's 40 atoms, it's near perfect, it's 4 billion years old. And how did nature ever manage to do this kind of thing? And then I'm going to show you something like really stunning. This is a gross hack. So one thing to realize is that DNA is redundant. So here we have a DNA string called TACGA. And as I told you, the T always pairs with an A and the C always pairs with a G. And you can see that these are the two strands of DNA, and this redundancy is what makes DNA so stable. So you can imagine that if you want to copy this, you could simply break up the DNA string and wait for it to repair itself. But the key insight here is that you see that the DNA is not quite symmetrical. So the parts on the left are different from the parts of the right. So DNA is actually point symmetrical. So if you want to read DNA, you can read it from the 5 direction to the 3 end. And that is in different directions on both sides. That's a key insight. Now, if you want to copy DNA, you could literally just tear it up and wait for the repair process to fix both of your DNA strands, so you end up with 2 DNA strands. Okay, that's a lot of fun. On top of the screen, you see all these spare DNA letters lying around, and they will now swoop in to repair the DNA, so we get 2 copies. And that's like really nice work. So actually multiplying DNA really is a matter of tearing it up and waiting for it to repair itself. But here's where it gets very interesting. And you have to realize this is a 4 billion year old software project. And if you've ever looked at like a 10-year-old software project, you realize it's full of gross hacks. So we can imagine that life would also accumulate a hack here and there. Now, I noted that DNA can be read in 2 directions and that it's not quite symmetrical. That means that to repair the left side of the DNA and the right side of the DNA is actually different work. That means that nature would have to come up with 2 DNA repair algorithms, one that treats the one side and one that treats the other side. And nature has decided not to do that. Nature has decided to reuse, code reuse, which is nice, which we all like. And it says, look, we're going to take the part that repairs the DNA on the left, and we're also going to use that on the right, even though that DNA goes in a different direction. And it's extremely sad that nature took this way, but it has delivered to us this marvelous animation. So what you see here on the left of the screen, DNA comes in, and one part is copied easily. That's the part that streams out to the bottom of your screen. So DNA streams in from the left, and a copy of 1/2 streams to the bottom of the screen, and that's all easy and simple. But since nature refused to evolve a part of DNA that could also do the copying of the other side, there is now a tremendously difficult process going on the top of your screen, where you see that the other half of DNA is being copied in reverse order. So it's sort of dragging the DNA back so that it can reuse the copying stuff that it uses on the other strand. And then it has to reassemble the whole DNA because half of it has been copied in the wrong direction. Now, this is like a really boneheaded software development thing where you would say, don't do it like this. Nature has done it like this. And this marvelous process you see going on here with DNS being copied in this haphazard way is going on within all forms of life, within us right now. Every one of us is performing billions and billions of these operations as we speak. And this is one of the sort of proofs that I think that life is really a software project where people can be supremely boneheaded and do the wrong thing for billions of years. But anyhow, this is what we are stuck with. And this sort of reverse copying process has left imprints on all of DNA because the top half of the copying process is far more difficult and actually more error prone, and that has consequences. So we now head to the territory of the coronavirus. And this briefly is what it looks like. In the middle, you see this little string, and that is actually the source code of the virus, the RNA that is in there. RNA is a sort of short-lived form of DNA. And you can see that this virus is a little circle and has these pointy things on it, which we call the spike protein. And now I want you to mentally go back to January 2020, when the first people were falling ill mysteriously and unexplained, and we didn't know what it was. And everybody, of course, really wanted to know what it was. And in the next few slides, I'm going to take you through the steps that happened in Shanghai at Fudan University. It's very pretty, and this is one of the premier most important universities in China, and it also hosts the Shanghai Public Health Institute, and they were the people that were doing the initial DNA analysis of the coronavirus. Even they didn't know it was a coronavirus at the time. They knew it as the Wuhan fish market pneumonia. So what happened over there? They started with this machine, and this is a very lucky accident. This is a DNA sequencer machine, and because it's such a messy photo, you know it was taken by me. This was actually the DNA sequencer that I worked with at Delft University, and it looks a lot like sort of an inkjet printer, sort of a big inkjet printer, and the parallels go deep. You put in the DNA in the left arrow, and there on the right there's a USB stick, and that's where the DNA comes out as computer files. It looks a lot like an inkjet printer, and the business model is the same. You buy this machine for like 50,000 euros or whatever, 40,000 pounds. And then it starts the consumables, the cartridges that go in. They are extremely more expensive than the machine itself. And there's DRM on them to prevent you from refilling them. So it's all terrible. But this is the workhorse machine of viral discovery, because this machine is affordable and it fits on your desk, and it doesn't require a whole laboratory. And so this is what they used in Shanghai, and this is also the machine I used. So how does that work? Here we have a bit of viral RNA that is converted to DNA. And we would love, of course, to read this whole bit of DNA in one go, so we knew what was going on. And sadly, we cannot do that. There is not really a machine in which you can take a whole string of DNA and that the machine will read the whole string in one go. We're still working on it. There's a company in Oxford Nanopore, that is really good at reading longer stretches of DNA. But for now, we cannot just read a whole string of DNA in one go. What can we do? We can take some material from an infected patient that is presumably full of viral RNA, convert it to DNA, and make a lot of copies of it. And you see this hot spring in the left, this very colorful hot spring. In that hot spring lives a bacterium called Thermo Aquarius, or TAC, as we call it lovingly. And TAC is able to really quickly copy large amounts of DNA at high temperature. So we use an enzyme that we got from that bacterium to make loads and loads and loads of copies of DNA. These copies are then sheared. So they're cut in many pieces. And so now we have lots of short bits of DNA floating around. And these are sort of things that the machine can start reading. But actually, the machine can only read a small part of these strings of DNA. And you can compare this process by taking a piece of paper, photocopying it 1,000 times, and then shredding those 1,000 copies with a really good paper shredder, and then handing you the bag with all the shards in it, all the little pieces of paper, and then giving you the challenge, can you reconstruct the original paper from all these shredded pieces of paper that I now gave you? And this is not hopeless, because there are so many copies of the paper. We know that some of these little shreds will overlap. So it's a possible puzzle. Okay, now imagine we put the stuff in this MySec DNA sequencer, and then this terrible file comes out. And biologists should not be let anywhere near computers, because they come up with stuff like this. This is 46 gigabytes of output from a DNA sequencer. And so it's a lot of data. And if you sequence a human being, easily 1 terabyte of this comes out. Why is it 1 terabyte? Because every read that comes from the machine is 4 lines, and all these lines keep on repeating themselves. So every read gives you the full serial number of the machine that was used to make it. So we are making millions and millions and billions of reads, and for every read we repeat the serial number of the machine that made it. And there's really no good reason for it. And then to encode, so we see lots of DNA in here. So we see actually CAC, TAC, AA. So that's the bits that we care about. And then we see a plus sign. That plus sign is there purely for historical reasons. There's no reason for that plus sign to be there. And then we see the line that starts with the hash, the pound sign. That is the quality. That is where the machine says for every bit of DNA read how sure it is that it got it right. And they encode this stuff as ASCII. And this is why even a small virus leads to 64 gigabytes of ASCII on disk, and it's 2020 and they haven't gotten around to really fix this. The way they fix this now, and I'm really upset about this, is instead of fixing the file format, they have now created a gzipped mounting construct where they can mount compressed data, so they don't have to fix the actual file format. So anyhow, it's upsetting. So these are like when you shred that paper, the 1,000 copies of that piece of paper you made, and you shredded all of them. And each of these DNA lines is like one part of the shredded whole. And the question now is, could we assemble from all these small viral parts, could we assemble the whole again? And actually, it turns out that even with the trusty tool grep that we use a lot, we can take a bit of DNA and just grep for that in the output of the sequencer. And here you see that in many of the reads, many of the bits of DNA, we find this random stretch of DNA that I selected. So we actually have proof here that the output of our shredding machine, that many of the small pieces of paper actually overlap, and because they contain the same stuff, the same DNA. And now we can align that. And that looks like this. We take all the DNA reads we saw on the previous slide, and we align them so that the common bits are the same. And from here, you can see that we've puzzled together quite a bit of the coronavirus DNA. So this is fun. Of course, no one should do this by eye because we have 64 gigabytes or 46 gigabytes of this stuff. So we use a tool called MegaHit. And you can get MegaHit from GitHub. This is the actual tool that was used in Shanghai to create the first copy of the viral, the genetic material of the coronavirus. And it's a lovely tool, and it probes how much memory your system has, and then it decides to use all of that, which is quite a rough surprise if you were also doing other things on your computer, because it just counts how much hardware memory there is, and it uses all of it. So I nearly lost my presentation over this yesterday. And it goes to think, and it starts to align all these reads to see if it can fill out the puzzle to get to the original, to a string of DNA that belongs to the virus. And now you have to again think about the situation in Shanghai, where these people were sitting there and they were analyzing the output of the machine generated from materials gathered from a very sick patient, and the machine sits there and it's calculating and it's calculating, and it took hours and hours and hours, and everyone is waiting to figure out what will come out. What comes out is this. The sequence MN908947.3 Wuhan seafood market pneumonia virus isolate. This is actually a file that ends up on disk. And actually, when they ran this analysis, lots of other files also came out because they were just sequencing genetic material from a very sick patient. But they knew this was the most prominently present part of that material. And they probably knew that, yeah, this is the virus, the mystery virus. And this file is 7.5 kilobytes in size, so it's reasonably small. And then I wasn't in the room, but I can almost guarantee you that this is what happened. You look at the file, and I've selected one line of this file, and then you go to a website called Blast. So I copy pasted this one line that's in green, and you paste it in Blast. And this is the Google of DNA sequences. This is where you can paste a DNA sequence, and it will find 100% identical matches, but it will also find fuzzy matches and very fuzzy matches. And I'm 99% sure that this is the first thing they did, paste in a bit of the viral DNA into the Blast website. And then what greeted them was this. You now have to wait, because sometimes this site is instantaneous, and sometimes mysteriously it takes many minutes to fulfill your search request. So everyone was probably looking at the screen right now and going like, get on with it, blast. And then this must have greeted them. And bad SARS-like coronavirus. And on the right in Chinese, I am told that this is Chinese for sharp intake of breath. This is the moment that the people in the room knew that the news was extremely bad, because the website told them this is SARS. And I cannot imagine what it must have felt like to be in that room, but this is what they saw, or something at least very close to it. If you redo this search today, you will get 1 million copies of the actual SARS coronavirus. So to recreate this experience, I had to exclude SARS from the search so that you can experience the full horror again. So that's what they did. Now, how does this stuff work? DNA gets converted into RNA, gets converted into proteins, and these proteins operate by the rules of nature. And this is very close to a hard drive that the data gets read into RAM, and that gets fed into the CPU. And the CPU is then actually a sort of physics project that works with electrons that obey the rules of nature. So this is a very close parallel. This is not a reach, this is not a stretch, this is actually how things work. And I'm going to skip this part, but you can see it later in the presentation. This is the ribosome, and this is the machine that converts RNA material into proteins, and it is glorious. If you have a 3D printer at home, then maybe it has a resolution of like, I don't know, 1 millimeter. I don't really know how that works. This thing prints by 20 atoms at a time. So it's extremely impressive. Everything in nature you see came out of this ribosome. And so we were built by ribosomes. If you are sitting at the table, it was built by a ribosome. This really is where everything comes from. Let's see if I can skip to the next slide. Yeah. So here's a table. Here's a table that says if you take the following bits of DNA, then you turn them into this is how you turn it into the output of the 3D printer. So you take 3 letters of DNA, GCT, for example, and that will lead the 3D printer to print an alanine protein. Or you can read AAT, and it will create aspartame, I think. And so each of these 3 letters, and it's important to realize this, DNA in this case always goes by 3 letters. You take 3 letters, and those 3 letters determine what the 3D printer, the ribosome, will do next. So it's always 3 letters. This is again what the coronavirus looks like, and these are the components. So we can see the famous spike S protein, we see the membrane glycoprotein, we see the small envelope, and we see the nucleocapsid. So every part of this virus is actually a protein and is therefore also a gene. And if you want to create a vaccine, we really need to know which part is what. This is what a viral genome looks like. It really says these are the various genes. And you can see a comparison between the original SARS from 2003 and MERS, which is the camel variant from 2012. And finally, below, you see the current SARS. And you see they're all very, very similar, within the DNA laid out these different components. And that looks surprisingly a lot like a sort of IKEA manual how to assemble a virus, where one of the very key insights is that this manual also contains instructions for creating another copy of the manual. So this is unlike your IKEA Billy experience where you end up with a Billy. But if you follow the coronavirus manual, you end up not only with a Billy, but also with a new copy of a manual how to make a Billy. And this is the literal viral part, how viruses manage to copy themselves into the trillions and trillions of copies, because they contain a full copy of making the manual that also instructs a cell to make another copy. So remember this file. This is the file they ended up with, the Wuhan seafood market pneumonia virus. And now what? Because this is just a whole bunch of letters. And we don't want a whole bunch of letters. We want to get the actual genes out of there so that we can figure out how this virus is made. So this is what a virus, what a gene looks like. And the important bits are with the gray arrows, the start codon and the stop codon. So this says a gene always starts with a start codon and it ends on the stop codon. What is a codon? A codon is 3 DNA characters. And in nature, the start codon is always ATG, and there are 3 stop codons, TAG, TAA, and TGA. And you know that a gene will consist of such a start codon, then a whole bunch of further codons, so further groupings of 3 DNA letters, and finally there will be a stop codon. Now, how could we isolate these genes from the file we just got? And you know the answer already, of course. Regular expressions. Now, this is, of course, not what people do in real life, but for this demonstration purpose, it works really well because finally we can use regular expressions for something useful. And at the top of the screen, you can see a regular expression that matches a gene. So how does it work? It says it starts with ATG. Then we need 3 DNA characters. And finally, we need it to end on TAG, TAA, or TGA. And surprisingly enough, if you let loose this regular expression, which is, by the way, a Perl compatible regular expressions for reasons, it actually manages to isolate a whole bunch of genes from the coronavirus genome, and including in the red circle, the famous spike protein. So this silly regular expression actually worked in isolating the spike protein, which is the one that is in all the vaccines. So don't do this for real, by the way, because there are far better ways of doing this, but it's sort of fun that this regular expression does get you the right protein. Now, when they did that, they found that the 2003 SARS protein was extremely like the 2019 novel coronavirus S protein. And this is, of course, bad news because SARS was tremendously scary. It's also good news because it is so similar that everything we had learned over the past 17 years about the original SARS was useful for thinking about the new coronavirus. And actually, the original SARS S protein is only 1 amino acid longer than the one in the current coronavirus. It is that close. And this is one of the explanations why we could make the vaccine so quickly, because people had been studying just this protein for the past 17 years already. So this is the actual mRNA that is in the Pfizer-BioNTech vaccine. And what you see in here is a sequence of RNA that is in the vaccine. And if you look carefully in bold, you see this A, Psi, G, Psi, P, C stuff. That is actually literally the amino acids, the same amino acids that we extracted with our regular expression. And they treated it a bit to improve the expression, as it's called. And I cannot go into that in this presentation, but our regular expression actually dug up the right RNA to put in the vaccine. And I still find this quite impressive. So what did people do then? They got this S protein and they knew if we show our bodies the S protein from the virus, our immune system will do the right thing. So now the goal is, can we actually make enough of this material to put into the vaccines? And they started with a machine like this. It's probably not actually one like this. This is the inverse of the MySac machine I showed you. In the MySac machine, you put living material and it creates a file for you with the DNA of that living material. This machine is the exact reverse. You can literally copy paste bits of DNA into here and then it will print DNA for you. And this machine is even more expensive than the other one to run. And it creates the tiniest amounts of DNA. So with this machine, you actually print a small bit of DNA that you want to put in the vaccine. And then you need to get it copied. And then again, we look at nature. You put all that DNA into microbes, like the E. coli on the right. And the E. coli, you put it in a fermenting apparatus, and then it will start copying itself for a few weeks. And in the end, you have liters and liters of bacteria that have faithfully copied your your vaccine DNA. So E. coli is the kind of bacterium that makes you really ill at grills and barbecues. But in this case, you can be grateful that it lent itself to copying all this DNA material for us. But it's still DNA and we need RNA. So how we're going to do the conversion? By using this little fellow. This is actually a bacterial virus. And I know the pictures on the top right, they look like they're very hard to believe that a virus could actually look like this. And the news is, it really looks like this. These are viruses that infect bacteria. And on the right, you can see sort of actual electron microscope photos with electrons of this virus, and it really looks like this. So it looks like science fiction. It looks like something from a scary movie or whatever. But this is what these bacterial viruses look like. And one of these things these bacterial viruses can do really well is convert DNA into RNA. So we have tasked this little bacterial virus to do that for us, and that converts the vats full of DNA into RNA. Now, this is again sort of repetition of how these vaccines work. And I still find this to be completely mind-blowing. So the goal of a vaccine is to educate our immune system without us actually falling ill. So we would like to show our immune system that, hey, this is what a coronavirus looks like. And please learn from this, but please don't die or anything. And until very recently, the way we did this was to inject a dead or a sabotaged virus or just small parts of the virus and then anger our immune system a little bit and then hope that it would learn from that. And for some vaccines that work really well, like measles, and for others, like the flu vaccine, it really doesn't work all that well because our immune system is sort of clever and it recognizes it is not very impressed by a dead virus. The immune system really loves it when it really pays attention when something looks like a real living virus and then it really springs into action. So this old way of making vaccines sometimes worked, but not so very well, and also required billions of chicken eggs and also a lot of good luck. Whereas with the mRNA vaccine, what we have done is we have taken literally the most important part of the virus and removed all the rest. And then we inject our bodies with instructions for making that most important part of the virus. Our immune systems are completely fooled because they think, yeah, this is the real thing. It's a virus is being produced inside of me and I need to spring into action and I need to mount a full response. And that is what it does. But because we cleverly only included one part of the virus, one part of this IKEA manual, the vaccine does not actually make us ill. And you have to realize that this is a breakthrough that people have been working on for 20 years. So this is not something that we invented last year for the coronavirus. People have been working on this for decades. But just now, when the virus came along, this technology was ready to be used. So we are sort of extremely lucky, but also lucky in the sense of the more I practice, the luckier I get, because people have been working on this for 20 years. So a final important bit, you cannot just inject mRNA in people and then expect the 3D printer to get to work. And part of this 20-year process was finding a way of encapsulating the vaccine mRNA in a way that it would end up in our cells and get executed by the 3D printer. And the funny thing about this is that it uses a few lipids, so it's a little drop of fat, and it includes, for example, cholesterol. And key of that is that our many of our cells say, oh yeah, this is the kind of stuff we need, and these cells will willingly absorb these little fat droplets with the RNA in there that you see the 3 copies of that in the middle, and they get into our cells, and our cells faithfully executed mRNA, make a scary virus part, and our immune system reacts to that, and we get this 95% immunity. And well, here's what I said already. the key part here is that the cell generates loads and loads of these spike proteins. Our immune system gets mighty upset. And you have to realize that many people that get one of these mRNA vaccines, especially the second dose, it really does fool your body into action. And you might reserve a spot on the couch after your second vaccination, because it could be a bit rough. But it should be the kind of rough that you spend a day watching Netflix, and then a week or so later, your immune system has done its thing. So summarizing this torrent of information, can we really say that life is digital? And I really do think so, but you really have to realize that just as in computing, the ones and zeros, there are not actually ones and zeros in our computers. There are voltages, and these voltages, they move electrons, and these electrons, they jump across band gaps. And so in the end, even though things start as digital, they end up being very analog again. But that goes the same way for computers as for these mRNA things. But can we call the vaccine software? And yes, I definitely think we can. It is digital information that we inject into a system. The system then executes that code. And I cannot get any closer to a definition of software than that. Beyond the scope of this presentation, it turns out that you can actually make conditional RNA that only executes under some circumstances. And in this way, you can craft actual whole algorithms in there. And so the other thing that's sort of fun to realize is that in most of the injections, there's 30 micrograms of mRNA, and every mRNA letter weighs somewhere in the order of 10 to the minus 21 grams. And if you do the math on that, that implies that one single coronavirus inoculation is actually 14 petabytes of data. And since you get two of them, in total, you get like, well, in the neighborhood of 30 petabytes of mRNA injected. And I personally look forward to that very much. But of course, we have to realize that it's actually 13 trillion repetitions of the same small bit of mRNA. So it's not quite that you get 14 petabytes, unique petabytes injected, but it's still a lot. As some of you know, I wrote a big blog post about this stuff, which contains partially the same information, but partially also very different information. And if you want to know more, I kindly suggest going to the link I put here. Of course, it's very difficult to remember that link right now. So I have this final page, which has an easier to remember URL, berthub.eu/links-mRNA. And there you can find this presentation, which also has some notes in there with links to even more information. And with that, I hope I have not overwhelmed you. No, I'm sure I've overwhelmed you with way too many details. But I hope that this has been useful, and I'm anxious to hear if you have any questions. Thank you.
Oh, thank you so much, Bert, for this fantastic presentation. I don't know if you've overwhelmed us, but you have certainly boggled my mind. Oh. I've read your article several times, but I still thought this presentation was fantastic. So I see there's some questions in the chat. I'll ask people to pop them in the Q&A, and we can get to them in a structured way. And I might actually start by letting Petra jump in with a question or two that she might have.
Yeah, thank you so much for this lovely presentation. And I always thought I wasn't into computing at all, but after listening to you, I think maybe it's just not all biology and genes for me. This is a very interesting approach. And I think you're bridging two rather different disciplines, or what at least you at first glance think is two different disciplines, DNA and vaccine using computing terms. And this is, of course, a fascinating and interesting exercise. But what are your thoughts about how this is a potential of bringing these two disciplines together? I mean, in terms of research and collaborating, when you use and change a bit the language, can this make us move forward?
Yeah, so it's a very interesting question. So this blog post I wrote was read by like 1.5 million people. And it also reached, because of that, it reached a lot of actual professional biologists and geneticists, and many of them sent me email. And the interesting part was that the leading people in the field. So I got email from all the laboratories involved with the big vaccines, and the sort of leaders in the field. They said, yeah, this is like a really good way of seeing it. And one of them actually emailed. He says, I finally understand my computer now, because he interpreted it completely in the reverse of how I intended it. But the really good people, they said this is a valid way of looking at it, and it's very useful, and also the analogies, and it brings us somewhere. And then there were a lot of sort of people that were slightly less informed biologists, and they said, Bert, you cannot do this. You cannot treat this stuff as digital. But since the really impressive people thought there was some merit to it, I do think there is merit to it, because there are concepts in computing, like bit rot is a good example, that we also see in DNA, and that we as computer programmers are like, yeah, I see what's going on here. Whereas the biologists are still thinking a lot, and I don't blame them for this, but they are still thinking a lot in terms of molecules. And it's true, they are molecules, and we should never forget they are molecules, but they're also data. And so whole concepts like entropy and coherence length and bit rot, they apply really well. And I think, to finally answer your question, I do think this is a useful way, and that it will actually advance the state of the art.
Yeah, and you make a really good job, because I think they can be stubborn, all these.
Yeah, they can be.
So I'll throw a question, and I see there's a question in the chat, and I'll try to marry my question with the question by Samath Jaju. So you touched upon the side effects of the mRNA vaccine, and the difference between a traditional vaccine that has a half dead or defunct virus, while the mRNA vaccine, it's, if I understand it correctly, is it correct to say that it sort of programs your cells, or it gives instructions to your cells to mount the correct response? If spike protein, and if it sees that the cells are invaded, so to speak, then mount this particular response. So if this is a semi-correct way of describing it, I want to try to understand if there are differences in side effects. You touched upon that a little bit, but since a defunct virus actually kicks your immune system into action, is there something categorically you can say about how mRNA vaccines actually trigger different types of side effects compared to the traditional vaccines?
Yeah, so I have to be sort of super careful in answering this, because I'm not actually a biologist or a medical doctor, but there are two important things to note. That if you just inject a dead virus, then the body is usually not super interested in that. So to actually make our bodies work, we also inject some other stuff called an adjuvant. And this adjuvant is there to just tell our immune system that, yeah, look, I know you're not impressed by this dead virus, but you should be impressed by this other stuff I just injected into you. And that makes our immune system pretty angry, and then it starts looking for trouble. And when it starts looking for trouble, it finds the dead virus, and it goes like, oh, I'll take a look at this then for you. So that creates some kind of side effect, like a little bit of fever and that kind of stuff. The mRNA viruses in vaccines, they are actually sort of doing the real thing, and they are really making our cells look like they are infected with the virus. And not to scare you or anything, but the cells in which the vaccines enters, so the vaccine enters a number of the cells, it does not end well for those cells. They mount such a convincing viral response that our immune system says, okay, we're going to kill you. And that's actually good, because you can easily make new cells, so don't get too attached to individual cells. But it's actually sort of a brutal affair. But here is why I do not worry about that. If you compare the vaccine experience, which is time limited, it's location limited, it happens near the injection site, it does lots of things that the virus does, but it does it in a time limited way, and it does far less of it. And that means that the more worried you are about the virus, the more interested you should be in the vaccine. Because it sort of generates the side effects of the actual virus infection include death. And the side effects of even the mRNA vaccines include lounging on your couch for a day watching Netflix. And I think that's a pretty good trade-off. But so far, and now we know, because we've so many millions and millions and millions of people have been vaccinated, that it's like nothing compared to the actual virus.
Yeah, interesting. I'm going to pick up some of the questions in that vein. So from Samath Jaju, why does the mRNA injector not harm our immune system? It seems like we're in fact injecting it. So why are there no problems, so to speak?
Yeah. Well, that's actually how our immune system works. That's what it does. It fights bad things all day long. And you have to realize when you live, when you breathe, you lots and lots of viruses enter your body, and bacteria and other things. So your immune system is actually at work all day long, killing bad stuff. And now the only thing that really changes is that we focus it. So the immune system is not suddenly just waiting there and not doing anything until the vaccine comes along. The immune system is just doing what it's always doing, and now it's doing it on this vaccine. But in the same way, whenever you eat something, you ingest. I mean, don't think too much about it. If you think too much about this, you will never eat or breathe again. But anytime you eat something, you eat billions of bacteria and trillions of bacterial viruses, and these are all dealt with. So it's nothing special, and it will not harm the immune system. It will only educate the immune system.
And in fact, if I understand it correctly, the traditional vaccines, they actually trigger, it works with the acquired immune system, while in fact these mRNA vaccines, they work with your innate.
That's not quite how it works.
Right.
But a very good difference to realize is that the sort of traditional vaccines, they get made by chickens, in chicken eggs, and or in insects. And then we try very hard to strip out all the egg and insect bits before we put them into a vial to inject it into you. And that doesn't quite work. It doesn't quite work. So in a traditional vaccine, there are always a few bits of chicken material present. And there are people that have chicken allergies or egg allergies, and they respond to that in a major way. Whereas the mRNA vaccines, they are like laser-like focused, and no chicken was involved with that. And that helps a lot.
All right. So we should advertise it as no chickens were harmed in the production.
Yeah, it's vegan. So.
Right. I see there are lots of questions. I do want to pick up one of your questions from Peter, because I know that she's got some good thoughts around this, and then I will definitely pick up the questions from the Q&A.
Oh.
It's a lot of discussions now about the mutations of the virus, and worries about that the current vaccines won't work if the virus mutate too much. And from your point of view, I think, because listening to you about the computing terms, is it more easy to just change a bit of information in the vaccine process to handle also the mutations? Can that be a quicker process than if you use all the chickens and the eggs and the good luck?
Yeah. So actually, this is sort of, you remember these Chinese scientists sitting behind their computer and being all scared that they just saw the first bits of the DNA. There's also the converse of that. There is now a file on a computer within Pfizer and BioNTech, which says vaccine.fasta. And someone can open that file in Notepad and change a few characters, go to the 3D printer, and a new vaccine comes out. And from the production side of things, it's actually completely trivial to change. So they could just say, okay, we just put the South African variant in there, and the Kent variant, and the Brazilian variant. We're just all gonna copy paste them in there. And from what I understand, it would take around 2 weeks from the change to the first new vaccine that you could inject. Now, there are other reasons why this process is now slated. The first suggestion is that the updated vaccines will arrive in September. But that is not in any way a technical limitation. That's just a regulatory limitation. But if they wanted, they could make a new vaccine every 2 weeks. And this, by the way, is also extremely exciting for the flu vaccine, which is usually 6 months late. So they have to guess what the flu, if flu ever comes back, because it has disappeared, and I'm not missing it, by the way. But if it ever comes back, we have to 6 months in advance. They have to think about what vaccines to make. Well, we could actually make a vaccine of the week right now. So
I think also this will affect the regulation process, as you.
Yeah. But it turns out that
big randomized tests.
Yeah, but it turns out that they also actually do not perform these randomized tests for updated flu vaccines, except for phase 1, phase 2. So of course, these variants are extremely worrying, and I'm not happy about that. But from a technical perspective, I mean, it's not that hard to open Notepad and make these changes. I mean, just don't make a typo. I mean, you should realize that, let's say you accidentally erase one letter, then you get a so-called frame shift in the DNA, and that you get an absolutely useless vaccine that is still very expensive and does nothing. So I assume they check it a few times before they get going on it.
Right. I think actually, in fact, you semi at least answered some of the questions in the Q&A. So I saw that Job Nijenhuis was asking, do you think it's possible to eventually use the regexes with mRNA vaccines effectively treating all kinds of coronaviruses in one go?
Yeah, so, I just butt in there. People are already working on the so-called pan-corona vaccine because all these coronaviruses have a common infrastructure, and which you saw in one of the slides. And it may be possible to create a sort of vaccine that is not, that is sort of an amalgam of these various coronaviruses out there. And it has already been done in a trial, and at least in that trial, they could make a pan-corona antibody that simply attacks COVID-19 and 20 and 21 and 22. So yeah, this is really a whole new world that's opening up.
Yeah, fantastic. I saw that Tony Yates was asking that too, about what's next, the common cold. And Will Hargraves asking, so where do you think we'll be in 20 years from now? Is it possible that we'll just go to our doctor's surgery to receive the latest freshly 3D printed vaccine sent over the internet that morning?
You know, predicting the future is very hard. But I've not always been good at that. However, very interesting, these mRNA vaccines were not actually made for this. The past 20 years, people realized that they could do far bigger things with mRNA. And so right now there is a BioNTech, the same firm that makes the big Pfizer vaccine, has created an individualized multiple sclerosis vaccine, which is not something you would traditionally see as a vaccine, but they've created mRNA that will calm our immune system. And BioNTech was actually not founded to make corona vaccines. They were founded to fight cancer. Because with this, you can also sequence a tumor, figure out the bad parts of it, then highlight these to our immune system, make an individual vaccine that is just tailored to just one tumor. So I think what really happened, to put this in a sort of historical context, when HIV/AIDS arrived, molecular biology was literally, not literally, but was like figuratively in the Stone Age. Because of HIV, because it impacted rich white people, we suddenly got interested in it, much more than fighting other diseases. So suddenly everyone turned into molecular biology, and HIV/AIDS was a breakthrough because we learned so much, and suddenly we were able to make all these interesting medicines. The coronavirus has definitely done the same thing over the past year. All kinds of techniques that had not left the lab because they were waiting for their big moment, they now had their big moment. So we're going to see this as a watershed moment for many things. And what exactly will happen, I don't know, but it will be a lot.
That's amazing. I mean, so in fact, you've answered one of the questions that I had in my head about if this, the discoveries have actually opened up possibilities to treat or prevent other types of diseases. So you mentioned MS, you mentioned cancer, and other particular types of diseases where this new technique, or so to speak, are particularly suitable? Or
yeah, so here I just have to say I don't know. Right. I will leave this to actual experts. I mentioned a few of the examples that I'm aware of. The cancer one I'm very excited about. Lots of people ask, can we now also do common cold? And the issue there is that the common cold is actually sort of 25 different viruses. So we cannot just go out there and make a common cold vaccine, and the time really will tell. But it is very exciting. Let me put it that way.
Yeah, fantastic. So Petra, I don't know if you wanted to jump in. I know that you had some questions that were maybe not as optimistic, or
no, but I think this is what I thought about. If the mRNA vaccines are to be looked at as a code, what do you think are the potential risks of creating a malware mRNA vaccine? That if somebody, like a bad person,
Yes.
now it's a sci-fi movie, but that you can do something that when it enters the body, it can activate, maybe not at once, but if your body is exposed to a stimuli and maybe in 1 year's time, a smell or a high temperature, then it will start to do something not so good.
Yeah. So let me share a little thing with you. When molecular biologists get together, and they no longer get together, but then they used to get together and drink beer. At some point, the discussion always turns to, we could do terrible, terrible things with the things that we just have lying around in the lab right now. And it doesn't bear thinking what an evil-minded person could do. And actually, some of the earliest discoverers of DNA were aware of this. One of them even left the field because he said this technology is too dangerous. And I don't think this is the specific breakthrough. There are other things I am far more worried about in the same field. But it is a double-edged sword. And the problem is we cannot put this knowledge back into the bottle.
No, it's like all the CRISPR techniques. And
it has escaped, and short of saying to everyone, we're going to destroy your lab and break your files and take away your computers, we're not going to stop this. And so for now, let's hope that we continue to do good things with it.
Yeah.
And I think, I'm sort of hopeful. But again, I have been in these meetings with actual biologists and microbiologists, and they go like, you don't know what I can do, and let's not.
Yeah,
I guess it's like any new technology, that it's about what ethical frameworks you create around them and what regulation and
but then of course, and we should really end this on a sort of happier note, because otherwise we start discussing what North Korea could do or whatever, and it's just too terrible to think about. So for now, let us also ponder that this offers great ways of reducing cancer and MS and other diseases, and let's hope we do the right thing. And for now, there's no going back. So let's think about the end of flu. I think that would be very good. Let's think about that we can reopen our societies thanks to science once we get all this stuff distributed to everyone, which is the big important part. But if we get that done, this will have been science turning around a pandemic in incredibly short order, which is nothing short of science fiction.
The bad stuff to the filmmakers, I think, or to Netflix creators.
Yeah. That is certainly an aspect of it.
Just have one more question, because I think it was very interesting that you said life is digital. It's the digital also alive?
Well, sometimes you do see these analogies. Sometimes you do see these things that happen. We have not yet been very good at evolving code in computers. Although, for example, if you try to create a neural network, you are in a real sense evolving a neural network so that it becomes smarter. We have seen, and that's a great example, comes from Google, where they created all kinds of computers that monitored each other and would distribute the load of the visitors among different computers using algorithms. And they found rather quickly that the system developed behaviors that they hadn't anticipated. So the system sort of figured out itself that it could balance the loads across continents in a way that the designers had not thought up before. So sometimes we do see these things that if you make your computers complex enough, they will start showing sort of behavior And I wouldn't say you would call it alive, but you would at least call it, say, well, the system responded to something in a way that I had not put into it. But I'm definitely sure that we're going to see these worlds converge further.
Yeah, for sure. So I'm conscious of the fact that we've reached the top of the hour, but I can also see that there are a lot of questions. I will allow people to leave if they want to, but before we finish, I'd like to actually finish on that sort of note, looking forward of what we can expect from this. So I see there's a question from Michel Timmers about, that the mRNA vaccines were developed very quickly, but the testing and approval processes took time. Do you think we can see those processes being shortened and those things happening quicker in the future? And then I also saw a question from Dinesh Babute about, with the possibility of these vaccines being developed quickly, does this also mean that personalized medicine is on the foreseeable horizon?
Yeah. So let's start with the first one. So I'm not an expert in regulatory things, but I do know that one of the people that worked at one of the vaccine companies, he injected himself with the vaccine in February. Because he was one of the people working there, and he was really afraid of the coronavirus. So he tried it on himself, and it turned into a huge red pimple because the vaccine wasn't very pure at that point. And but it is, I mean, people could have been vaccinating themselves in February already, and some of the there were people that were working on it themselves that thought it was a reasonable thing to do. So could you speed this up? I can only speculate a little bit. We have two ways of evaluating if a vaccine works. One of them is actually multiple ways, but one of them, really quickly. We can inject an animal or a volunteer with the vaccine, and we can measure in their blood easily within 2 weeks if antibodies have been generated. So you can very quickly figure out on paper if it works. But it does not mean that I will actually work outside of the lab. But these are called markers, and the hunt is now on for biomarkers where we can say, look, this vaccine is going to work. We can validate in the lab with a person or with a monkey often that it actually works. And we have confidence that this will translate to real life. And I'm pretty sure, I know that people they have already done this process for the updated vaccines that have been made for the South African variants, where they say, look, we know that works. At least it works in the lab. Getting to the niche question, and it's very good that you're here, Dinesh. Yes, this is entirely possible. And if you look at the cancer vaccines that BioNTech is really very big in already, which was, like I said, their original game. Oh, that's just your vaccine. Well, I hope it's not your vaccine, but should someone ever fall ill, you get a vaccine with your name on it. It just says, look, this one is only for you. And it will, in fact, if you give it to someone else, it will do nothing. So it will be that individual. You might get your own vial. And this is entirely not science fiction. This is stuff that is going on right now, and that is actually working. And I'm very impressed by the BioNTech people that they have managed the coronavirus is done. We're now moving on to the next things already, and they're doing that. So yeah, that's going on.
That's mind-boggling. I believe that we have reached the top of the more than top of the hour, and we will have to end it here. And I apologize to all of you who have questions in the chat that haven't been answered. This will be published on YouTube afterwards, so maybe you can ask the comments in the questions in the comments.
Yeah, or you can also find me on Twitter.
Yeah.
And I'll just stick around for a while. So if you hit me up on Twitter, I'll just see if I can respond there.
Fantastic. Yeah, that's a great way of doing it. So with that, I want to say then thank you for this absolutely mind-boggling presentation, Bert. It was a real pleasure listening to it, and I think it opened up a lot of interesting thoughts in many of us, whether you're in the medical field or in computer science. I think it's raised a lot of interesting questions in our head. And this presentation will be available on YouTube afterwards. And I might even watch it again afterwards to see what new things I can learn from it. So thank you so much, Bert, for this amazing presentation.
Okay, well, thank you for the opportunity to do this. And I wanted to show it from another angle than I did in my earlier blog post. And doing that, I also learned a lot of stuff about the DNA processing. So it was a very rewarding process for me as well. So thank you very much.
That's great. And I think I'd recommend all of you actually to watch. I know Bert has given presentations on DNA and similar things on that's available on YouTube. So do a little search for Bert Hubert on YouTube and you'll find a bunch more fantastic presentations.
And I will definitely spread this to my medical community. I think they can learn a lot. Thank you so much.
Okay, thank you.
And thank you, Peter, as well, for your great questions. And thanks to all of you who participated to make this webinar an interesting one. I do want to say, keep an eye out on the LINX website for upcoming presentations and tutorials. We do a lot of technical tutorials as well as these LINX Distinguished Speaker Series presentations. We have some really interesting presentations coming up. So keep an eye out and make sure to register for those as well. And you'll be able to watch any past talks on the LINX website and the YouTube channel. So with that, I'd like to again give a very special thank you to Bert for taking the time to share his fantastic presentation. If you haven't read his original article, do make sure to do that as well. And do like I did, force all your friends and family to read it and then question them, interrogate them and see if they've understood it. It's been an absolute pleasure listening to you. So with that, I want to say thanks to all. Be well, stay healthy and safe, and hope to see you all very soon again.